Hello, I'm Stephen Harrison, a visiting professor of hepatology at the Radcliffe Department of Medicine, University of Oxford, the medical director of Pinnacle Clinical Research, and the president of Summit Clinical Research. It is my distinct privilege and honor today to talk to you about NASH treatment in 2022. To understand where we are with NASH therapy, we must take a brief tour through the pathogenesis of this disease and understand that there are multiple different pathways by which patients develop underlying steatohepatitis. There are three main ways that disease presents in these patients. Number one, insulin resistance. All of these patients are insulin resistant and or have diabetes. What happens with insulin resistance is adipocytes become dysfunctional and they release free fatty acids that are very toxic to the liver. They're taken up in the portal vein and delivered to the liver, where the liver has a couple options. Those toxic free fatty acids can be esterified to triglyceride and stored as fat. They can be burned through beta oxidation within the mitochondria, or they can be repackaged and shipped out of the liver in the form of very low density lipoprotein. Additionally, in patients with underlying non-alcoholic fatty liver disease and non-alcoholic steatohepatitis, there's a disproportional amount of upregulated de novo lipogenesis. And this provides another opportunity for target therapeutics. So in addition, as livers become full of toxic free fatty acids, mitochondrial dysfunction begins to take place. Endoplasmic reticulum stress, an increase in the unfolded protein response, ensues and drives pro-inflammatory mediators accentuated by the development of reactive oxygen species as well. This leads to apoptosis and subsequent activation of hepatic stellate cells. There's also an overlay of dysfunctional regulation with the immune cell pathways, and that can also contribute. In addition, the microbiome may be playing a role here as well, and genetic influences such as HSD17 beta 13 and PNPLA3 may also be contributing to underlying pathogenesis of disease. Now, all of these provide opportunities for drug targets. Many you see listed here, and I've broken them down into metabolic, anti-inflammatory, and anti-fibrotic. The majority of therapies being developed today for NASH target metabolic processes, as you see illustrated via FXR agonism, FGF19, FGF21 agonist, PPAR agonist, thyroid hormone receptor beta agonist, GLP-1 receptor agonist, FASN inhibitors, and acetyl-CoA carboxylase inhibitors. This is where we see the majority of work being done today and where we'll focus our energies. However, it does not mean that there are not other pathways on the right targeting anti-inflammatory therapeutics and direct anti-fibrotic therapeutics. There are those being developed too, just not quite as many. And ultimately, we are likely going to use combination therapy where we'll target multiple of these different pathways in our treatment of underlying NASH. So just to drill down on what I mentioned a little bit further, there are really 5 different buckets we could put drug therapies in. Targets related to insulin resistance and or lipid metabolism, targets related to lipotoxicity and oxidative stress, targets related to inflammation and immune cell activation, and then targeting stellate cells directly or targeting cells that are undergoing apoptosis or necrosis. I've highlighted in the next slide all of the treatments currently in development, with a special highlight in red for those that are currently in phase 3. And as you can see, the majority again are in the first 2 buckets, with some development being done on fibrosis and a few with immune cell activation and targets related to cell death. It's also important to note that some of these drugs have pleiotropic effects and may occur in multiple different buckets. Now, I'd like to take this opportunity to explore the drugs in phase 2. Now, we can break this down into phase 2A on the left and phase 2B on the right. The phase 2A trials are generally shorter duration, and in addition, they're non-invasive, meaning they don't do histopathology. And you see a myriad of drugs currently in development. On the right, even more drugs have made it into phase 2B. Here, it requires paired liver biopsies generally, a biopsy to get in and a biopsy to get out. Also, generally, these trials are larger in scope and longer in duration. Now, in the box shaded green, I've highlighted the drugs that are currently ongoing in phase 2B. Using the arrow, these are drugs that have either completed enrollment or have read out the results. So as you can see, there is a tremendous amount of activity underway right now in this field, and 2022 promises to report out numerous of these drugs, and we're hopeful that they will have positive results. Now, we can also break the phase 2 treatments down into those that are oral agents and those that are injectable or infusions. I want to focus on the right, injections and infusions. So if you are treating a patient with a hormone, there is no oral therapy for that. It has to be injected. And that's what we see with FGF19 and FGF21. There's only one FGF19 analog, that's NGM's aldafermin, and that is currently in a phase 2B trial with well-compensated cirrhosis. There are multiple drugs, multiple companies studying FGF21 agonists. In fact, many are well underway with enrollment, and it is anticipated that Akero will read out their 24-week paired liver biopsy study later this year. When looking at the GLP-1 receptor agonist, I not only put GLP-1s alone, but also those in combination with either GIP, such as tirzepatide, or glucagon agonists, such as cadutide or pemvidutide, and then a triple agonist by Hanmi. There's also a DGAT2 inhibitor. This is using antisense technology, and DGAT2 works with de novo lipogenesis to block de novo lipogenesis. And then finally, galectin-3 is the only drug in development right now that looks at prevention of progression of disease. Every one of these agents listed here, and that I will talk about subsequently, are targeting improvement of underlying histopathology or improvement in non-invasive technology. Galectin-3 inhibitor is an infusion given every 2 weeks. It is an adaptive phase 2B3 study right now. The idea being patients are enrolled with well-compensated cirrhosis who have no evidence of esophageal varices. And the goal of treatment is to prevent the progression of varices. Now, let's look at the drugs in phase 3. There are currently 5. However, one, Aramcol, is on hold. So we won't talk about that one more today. We'll spend the rest of our time talking about the 4 that are currently in development. There's an FXR agonist, a beta-cholic acid. There's a pan-PPAR agonist, lanifibranor, a thyroid hormone receptor beta agonist, resmetirom, and a GLP-1 receptor agonist, semaglutide. So first, let's look at drugs that have shown evidence of benefiting the patient from a fibrosis perspective. So here I've listed the drugs across the top: efruxifermin, lanifibranor, aldafermin, resmetirom, seladelpar, Aramcol, semaglutide, and Ocaliva, and then the dosing duration above that. And these bar graphs are looking at the proportion of subjects with at least 1 stage improvement in fibrosis and no worsening of the NAFLD activity score. Let's first look at those drugs that are in phase 2, and they're highlighted in green. So efruxifermin was dosed for 16 weeks, and we see 62% improvement in fibrosis with the high dose group, and 48% when you take all comers, compared to placebo with 0. Now, it's interesting to note the way this study was designed is patients only underwent liver biopsy if they had improvement in the non-invasive test, particularly MRI-PDFF. So not many people in placebo achieved that. In fact, only 2 underwent biopsy. So the numbers are relatively small. but it does show the potential impact of this class of drug on fibrosis. Now, aldafermin is an FGF19 analog, and we see a 20 percentage point treatment effect delta with 1 milligram versus placebo when treated for 24 weeks. When we focus on seladelpar, it's a PPAR delta agonist. It's an oral therapy. Study was for 36 weeks, and we see at the high dose group a 37% response on fibrosis with placebo at 20%. Now, this was not statistically significant, but it was a post hoc analysis and it was not powered for that endpoint. When looking at compounds that are now in phase 3 development, I show you results from their phase 2 trial. And here we have lanifibranor, 48% with the 1.2 gram dose or the 1200 milligram dose, compared to 29% for placebo. Resmetirom, 29% versus 24%. And I must say, parenthetically, with lanifibranor, the fibrosis treatment effect delta was significant. With resmetirom, it was not. And thus, with resmetirom, they're targeting predominantly NASH resolution in their phase 3. Aramcol, 29.5% for high dose versus 17.5% for placebo. Semaglutide, a bit all over the map, but you see a 32 to 49% treatment response rate with drug versus 33% for placebo. This was not statistically significant. And with Ocaliva, 23% versus 12%, the treatment effect delta of 11% was significant. And that was in the phase 3 trial. So everything I showed you up until Ocaliva was data from phase 2. The Ocaliva study was the Regenerate trial. That's the large phase 3 trial that led to the initial filing for the new drug application. Unfortunately, because the therapeutic index was deemed to not be adequate because of some adverse events linked to this drug, the FDA did not approve the drug. However, on July 7th, the company announced they are pursuing a refiling of this drug based on additional data they were able to obtain and additional safety data as well. So more to come as they begin the refiling process. Now let's look at NASH resolution. Again, using the same color coding for those drugs from phase 2, we see efruxifermin having a 54 to 48% response compared to placebo of 50%. But remember, only 2 patients in placebo underwent a biopsy. So some of these numbers might be conflated a bit. And I just think we need more data before we understand exactly how effective this particular drug, which is an FGF21 agonist, is on resolution of NASH. Additionally, treatment was only for 16 weeks. So additional asterisk there for that particular patient that did respond was that the patient lost 25 pounds or 11% weight reduction during the period. So it makes sense that there would be NASH resolution. Aldafermin, 24% versus 9%. Seladelpar, 26% versus 8%. When looking at those drugs now in phase 3, we see that there was a response with lanifibranor, 49% high dose versus 22%. That was significant. With resmetirom, 25 versus 15%, which was significant. Aramcol's on hold, but it did show a 16.7% with high dose versus 5%. And semaglutide, 59% with the 0.3 milligram dose daily injection versus 17% for placebo. And then those are phase 2. When we go to phase 3 with Ocaliva, you see a treatment effect of 12% with high dose versus 8% that was not statistically significant. Now, maybe a higher bar, absolutely a higher bar, is hitting both NASH resolution and fibrosis improvement. And we see data in phase 2 here with efruxifermin, aldafermin, and seladelpar. So first with efruxifermin, 28% versus 0%. Again, higher bar here. What you'll see consistently with this histopathologic endpoint is that it's harder for placebo to achieve success. And many people look at that as potentially an attractive target then because you're stabilizing placebo response rate. Yes, it's harder for drug to hit that as well, but it's potentially easier to see truth, ground truth. How well is your drug really working? Now with aldafermin, 22 versus 0, And then with seladelpar, 19.6 with high dose versus 8.3 with placebo, and a nice dose response relationship is seen. It's also important, again, to highlight the stabilization of placebo response rate. Now for phase 3, lanifibranor, we see 35, 25, and 9, so a nice dose response relationship. Again, placebo single digits. With semaglutide, 37% versus 15. A little bit higher placebo response rate there, maybe something we haven't seen with others when looking at this combination, but there was a higher response with semaglutide as well. Now, what about cirrhosis? Where are we at in drug development for cirrhosis? Well, we have 5 ongoing phase 2 or phase 3 trials. I have them all listed here. You have a beta-cholic acid, which is an FXR agonist, belapectin, which is that galectin-3 inhibitor I already mentioned. It's an infusion given every 2 weeks. That's a unique adaptive phase 2b3 study looking at the prevention of development of varices. Aldafermin, an FGF19 agonist that is in phase 2 with 150 patients, and that is fully enrolled, and we're just waiting the outcome. Efruxifermin is in phase 2. That's an FGF21 agonist, and that is also underway now with recruitment. And then finally, a combination study of semaglutide, which is a GLP-1 receptor agonist, selefexor, which is an FXR agonist, and firsocostat, which is an acetyl-CoA carboxylase inhibitor, is now also underway with enrollment in this well-compensated cirrhotic population. So let's briefly touch on when and where we anticipate these phase 3 trials reading out. As I mentioned already, Aramcol is on hold. Resmetirom is anticipating a readout, not Q3, shifted to the right there, but in Q4 of this year, with long-term outcomes anticipated sometime between Q2 and Q3 of 2024. A beta-cholic acid, I mentioned on July 7th this year, a press release came out that they are refiling based on new data and additional safety endpoints, additional safety data in many, many more patients. But that study is also pushing forward to an outcome that is anticipated in Q3 of 2025. So the refiling is based on the surrogate endpoint or conditional approvable endpoint of a liver biopsy at 18 months, but they still have the trial ongoing to look for overall outcomes. Semaglutide and lanifibranor are still enrolling with anticipated surrogate endpoint readout in Q3 of 2024 for semaglutide and Q2 2024 for lanifibranor. Now, what are some lessons learned along the way in drug development? Well, there are many trials that have ended in failure, but it doesn't mean we didn't learn from them. Many of the drugs you see listed here. What I would like to say is we need to begin to look at these drugs, not only for their histopathologic effect, but for their extrahepatic effect as well, and their tolerability profile. When we think of drugs, we think of therapeutic index. Therapeutic index is how effective the drug is versus how well it's tolerated. Now, I've broken this slide into 2 parts, non-cirrhotic on the left and cirrhotic on the right. And I've color-coded the drugs, red for mild adverse events and blue for mild to moderate adverse events. The ideal drug, in my opinion, would be a drug that's in the upper right-hand corner of this graph for both non-cirrhotic and cirrhotic. Let's start first with non-cirrhotic NASH. We see very good results on extrahepatic effects of drug with semaglutide, a GLP-1 receptor agonist, and efruxifermin, an FGF21 agonist. These drugs have shown beneficial effects not only in the liver, but also on glycemic control, atherogenic lipids, and weight loss for semaglutide. And for efruxifermin, weight is neutral to slightly down, with potential impacts also on blood pressure. Now, histopathologically, we see they're ranked kind of in the middle because the data sets are small for efruxifermin, and we don't fully yet know the potential impact on histology. But with semaglutide, there was no benefit seen in the large phase 2b trial on fibrosis, but a very nice improvement of NASH. Lanifibranor gets maybe slightly better nod on histology, but not quite as good on extrahepatic effects. It does have impacts on glycemic control and atherogenic lipids, but it also is linked to potential weight gain. When looking at the right with efruxifermin, that is the drug that we have maybe some data on. And I think it's important to note with cirrhosis, we're very limited in liver biopsy data. Certainly with belapectin, there is no histologic effect seen in the phase 2, and it is being studied to look at prevention of progression to varices. There is no extrahepatic benefit that we know of with belapectin. Alternatively, with efruxifermin, I've already listed the extrahepatic benefits. Where we're limited is just on the overall ponderance of evidence on histology in a well-compensated population of cirrhotic patients. There's only been a handful of patients studied, although in that study, a third of them had resolution of cirrhosis. So finally, we're at the Wright brothers stage of drug development. We're just getting to the point where we're taking off and able to have some response. Where we want to go is the F-35 precision medicine strike fighter approach, where we're honing in on therapeutics for patients that are targeted to their metabolic profile and to their genetic profile. And it's also important that we're likely to use combination therapy to get to this point. And when we think of combination therapy, we need to think of the risk tolerance, the safety, and where the patients are in their liver disease progression. For instance, if patients have very mild fibrosis, but have evidence of a lot of fat and inflammation, maybe a drug that is focused predominantly on improving metabolic profiles might be the backbone target, with an overlay of some fibrosis improvement with a second agent. Alternatively, if a patient has cirrhosis or advanced liver disease, we want to halt disease progression as quickly as we can. And so we want to target a drug that might have more antifibrotic effect and marry it up with a drug that potentially has some metabolic benefits as well, because we want to get their disease arrested or improve it, but also hit the drivers of fibrosis, blocking new formation of scar. So what is the ideal combination therapy? Well, in my opinion, it's a drug that's oral, well tolerated and safe, and essentially is able to combine therapies in a single pill. We want them to be synergistic, improving histology and improving extrahepatic metabolic profiles, with the long-term goal of improving outcomes, not only major adverse liver outcomes, but major adverse cardiac events as well. What would that be? Well, I illustrate this slide, which comes from a good friend of mine, Mike Charlton, where he showed this Jamaican 4x100 meter relay world record team that you see illustrated here. They set the world record in 2011 in Seoul. And a couple things to note. All of these individuals were world-class sprinters in their own right. They got along well with each other. They had complementary strengths, similar endurance, and from the same home. Ideally, combination therapy for NASH would hit 4 of these 5. World-class in their own right. They are harmonized and synergistic with each other, but they have complementary strengths, and they're meant for the long haul. In other words, we don't develop a situation where the drug no longer works in the patient over time because of anti-drug antibodies or some other reason, that they have endurance. So combination therapies that are currently underway are listed here. And I would just say that there are more and more of these coming forward. And this is what I'm very excited about for the future. When we think of combination therapies, we need to think of different buckets. Those that target overnutrition or endocrinopathies, those that focus on ER stress, lipotoxicity, or mitochondrial dysfunction, those that target inflammation, cholesterol toxicity, or are multifunctional, and those that target fibrosis. And ideally, we would pick drugs from each of these different buckets, or at least 2 different drugs from different buckets. Just as an example, I put in green hearts those that I think might be good combinations. So semaglutide on the left, lanifibranor, resmetirom in the middle, and maybe even obeticholic acid on the right, because it does have some fibrosis improvement qualities that I think would go well with, say, something like resmetirom, which is a thyroid hormone receptor beta agonist that has shown beneficial effects on steatosis improvement and NASH resolution, but less effects to date anyway on fibrosis. So putting that oral drug with the oral drug of beta-cholic acid, potentially we would synergize NASH resolution, fibrosis improvement. We would get additional benefits on atherogenic lipid improvement, and we would potentially be able to mitigate some of the adverse events of a beta-cholic acid by, in theory, using a lower dose of that drug when combined with resmetirom. Finally, future therapeutic considerations. I think as we move toward precision medicine, there are several things that we could do to target that. One is CAR T therapy, and I illustrate this by the mouse model on the right. So we historically think of CAR T therapy in cancer, but what about using it in an advanced cirrhotic population or fibrotic population? This is a very elegant study published in Nature in 2020 of an animal model with significant fibrosis, where CAR T therapy was utilized targeting senescent stellate cells. And by blocking these fibrosis-producing cells, you can see that there was resolution of scar over time. In addition to that, there was also improvement in liver chemistry tests. We're a long way to go before we can use this in humans, but it illustrates the principle that specific targeted precision medicine, getting after a specific cell type, might be advantageous and potentially used in combination therapy. Finally, therapeutic oligonucleotides provide another mechanism by which we could target therapies. I mentioned one already, and I want to highlight it again today. The DGAT2 antisense oligonucleotide is already in phase 2b therapy. It's an injectable. There's also another one targeting fibrosis, not NASH specifically, and that's heat shock protein 47. We could also target the microbiome as we begin to learn more and more about the microbiome and its influences in the setting of NASH. Genetic polymorphisms can also be evaluated and targeted, specifically those in development now, HSD17 beta 13, loss of sense mutation, and then PNP-LA3. With that, I would like to conclude my presentation and thank you for your attention.